it has been recording. Fine, let me share the screen then. Okay, I do think the slides are visible. If you cannot see the slides, let me know. I think it is visible. Okay, so let us start. So in today's class we will mainly discuss about the questions that are related to ATP synthesis. So regarding week 11 questions we will first discuss, and I think we will have another extra class that for my case it will be on 16th. It will be notified to all of you through email. So in that class we will discuss about the questions regarding week 12, as well as if you have any doubts throughout the course or some extra questions as well regarding the exams and everything. Okay, so without further ado, let's start today's session. Okay. So the first question that I'm going to show, I am not going to ask for an answer because it is a very easy one. So the question number 1 states, what is the ultimate source of energy for all living organisms? And you have your 4 options. So I am not asking the answer because I think all of you know this. So the options are very obvious: food, water, solar energy and chemical energy. For any of that to remain, the first thing that you need is solar energy. Without that, you will not be able to have any other things. So the ultimate source of energy for all living organisms is nothing but solar energy. It was just a general knowledge based question, so I will not spend much time in that. Okay, let's start with this one. So the second question states, the vitamin niacin is a part of which molecule? There is a blank that you need to fill. They are talking about vitamin niacin. You have to identify niacin is a part of which molecule. There are 4 options: ferredoxin, pyridoxal phosphate, pyrophosphate and NAD+. Out of these 4 options, choose your correct one and write it in the chat box, and then I will show you the correct answer. The vitamin niacin is a part of which one? Okay, I have one answer. Yes. So first let us look at the structure of what is this. This is your NAD+, nicotinamide adenine dinucleotide phosphate. This is the structure for NAD+, and if you remember from your last class, one second. If you remember your last class, we have discussed about niacin, that is basically your vitamin B3, where we discussed that how we can convert with what is this. It is nothing but your nicotinamide, which in presence of the cofactor niacin, it was being transferred to nicotinic acid was being transferred to nicotinamide for its action. That what we discussed in the last class. So yes, vitamin niacin is part of what? Nicotinamide adenine dinucleotide phosphate. Okay, so our correct option should have been NAD+. For any of you, if I have any doubt, we have our nicotinamide here, we have our adenyl group in the second part, and why it is dinucleotide? Because of this 2 phosphate groups that are attached with each other. So overall we have our nicotinamide adenine dinucleotide phosphate, where vitamin B3 takes part for conversion of nicotinic acid to nicotinamide, thereby forming this final nicotinamide moiety that you are seeing here. Okay, let us move to the next one. It states, which of the following statements about the chemiosmotic theory is correct? So we are talking about chemiosmotic theory. You have to identify, one second, I think it is showing, yes. Okay, you have to identify the correct statement that is explaining about chemiosmotic theory. Go through the statements, read them one by one, then select your correct option. The first one states electron transfer in mitochondria is accompanied by an asymmetric release of protons on one side of the inner mitochondrial membrane. The second statement says it predicts the oxidative phosphorylation can occur even in the absence of an intact inner mitochondrial membrane. Third one is the membrane ATP synthase has no significant role in the chemiosmotic theory. And the fourth one is the effect of uncoupling reagents are a consequence of their ability to carry electrons through membranes. These are the four statements. Which among them describes about chemiosmotic theory? Select your option and then obviously I will discuss. So let me just give you the hint if you are getting confused that what is chemiosmotic theory or what is chemiosmosis. That is the process of ATP synthesis using free energy obtained when electrons are passed to several carriers. That is the process otherwise known as chemoosmosis. Because we are reading about oxidative phosphorylation and the process of that is basically known as chemiosmosis, where many electrons are passed through several electron carriers. Okay, fine. So let me draw it here. So what was the thing that was happening in case of chemiosmosis? We had our mitochondria. We just draw it here. This is one of the cristae of mitochondria that I am showing. This is outer membrane. This one is your inner membrane. In between you have your intermembrane space. And inside you have your matrix. So this was the design what we are reading. So in the intermembrane position you have high concentration of H plus. That's why this section of the membrane will be positively charged, and in the matrix section you have less number of H plus. This will lead to a negative charge on this side of the membrane, and because of that when you require to synthesis ATP you require a transport of H plus ion from the matrix to the intermembrane space. That is coupled with the ATP synthesis, and the free energy that is required during this passing of this electron that are associated, that is known as chemiosmosis. So what should be your correct option then? I have shown you the process. I have described the describe the statement that what is chemiosmosis. So according to that, if you have to match chemiosmotic theory, in case of that, electron transfer in mitochondria is accompanied by an asymmetric release of protons on one side of the inner mitochondrial membrane. That is the correct statement. So in the matrix we have low concentration of H plus, and in the intermembrane segment we have high concentration of H plus. Still, for the ATP synthesis you require a transportation of H plus from this matrix side to intermembrane. That's why in the first statement it is written as asymmetric release, because otherwise what we know, whenever some ion concentration is greater on one side and less on another side, they will usually diffuse so that the concentration becomes equal. But in case of ATP synthesis we require this proton pump so that the charge gradient remains throughout the process. Only in that case we can gain the necessary free energy that is required for the ATP synthesis. So according to that, our chemiosmotic theory, the first statement is the correct one. Okay, I hope this one is clear. It was just talking about the process through which the proton transportation occurs. Fine. Let us move to the next one. So you have to match the entries in column 1 with those in column 2. Now in column 1 we have 4 things: vitamin B1, carboxypeptidase, TCA cycle and reducing sugar. And in column 2 we have 6 options. So carefully choose your that what will match with what option in column 2. Okay, so P will match with what, Q will with what, R with what and S with what. There will be 2 extra options left. So carefully match them and tell your answer in the chat box, and then I will discuss all those one by one. First try it out by yourself and match them according to your knowledge. Try to match them. At least you should all know about the vitamin B1. Just in the previous session we discussed about vitamin B1. Okay, we have one answer. Thank you. Let us look at that. So first coming to, let me show the options and then I will discuss. Okay, so first let's come to vitamin B1. What is this? It is thiamine basically, and in column 2 we can see the activated version of thiamine, that is thiamine pyrophosphate, TPP. About that we discussed in details in the last class. So vitamin B1 will match with thiamine pyrophosphate. So P will match with 1. So from that, very nicely at the beginning you can cross out option number B and D, because you know P matches with 1. Now we have to select between this A option or C option. Now look at them. In both the cases A and C, Q is matched with 4. So what do we have in Q? We have in Q our carboxypeptidase. What is the structure for carboxypeptidase? I have shown it here. And you can see in case of carboxypeptidase we have a zinc 2 plus that is associated with it. So carboxypeptide will match with this fourth option in column 2, that is zinc 2 plus. That is why Q is associated with 4. Coming to the third one, that is TCA cycle. Now in option A it is associated with aconitase, and in option C it is associated with riboflavin. So this is the image of TCA cycle, and the very right hand side image. In this TCA cycle what happens, it's the conversion of citrate to isocitrate, and that is being governed by citrate acetonitrase, citrate aconitase. Sorry for that misspelling. Okay, so this citrate to isocitrate conversion through aconitase happens in case of TCA cycle. So our TCA cycle with match with option number 2 in column 2. Okay, so R matches with 2. So we already have our correct answer, that should be option number A. We can already know that, but still look at the fourth one, that is reducing sugar. Now you have to identify among this option that which one of them is reducing sugar. So obviously riboflavin is not, it is not that. So sucrose and lactose, these are the two sugars that we need to think of. Now these are the structures that I have shown here for lactose and sucrose. Out of them, which one is are reducing sugar and which one is not. Lactose is a reducing sugar, this one, the left hand side one, but sucrose is not a reducing sugar. Why not? Because if you look here at this glycosidic linkage, what are these things? If you consider this carbon atom and this carbon atom, this is acetals. So the hemiacetal is gone from sucrose, because in that case glucose and fructose are attached together with this glycosidic bond, where you don't have your hemiacetals anymore, which can act as reducing sugars. But in case of lactose, you can see we have our hemiacetals present in both the carbons at these two positions. So obviously lactose will be able to act as a reducing sugar, but due to this glycosidic bond at this position, they will not be able to act as a reducing sugar. So in those options, S was corresponding to sucrose. So now we have our answer, P1, Q4, R was matching with 2 and S with 6. So the correct option should be option number A, as was said in the chat box. Okay. Let us move to the next question. Okay, fine. Which statement about FeS protein is incorrect? So we have our iron sulfur proteins as electron carriers. So among these four statements, you have to identify the incorrect one. Select your option and then I will discuss it. So there are four statements. First one is a rubredoxin contains a FeS4 units, where each S is coming from a cysteine residues. The next one is 2Fe2S, or you can call it also Fe2S2, that is ferrodoxin, contains 6 S donors, 2 of which are S2 minus ligands. Third one is a 4Fe4S ferrodoxin contains a Cuban cone, and fourth one also about a 4Fe4S ferrodoxin, that the 4 redox couples that makes use of the 4 epicenters are accessible in nature. These are the 4 statements. So let us first look at the first statement, that is A. Rubredoxin that contains a FeS4 units. So how does that look? In case of FeS4, we have our 4 sulfur atoms that are all coming from cysteine residues of your protein. So that is the structure for your FeS4 rubredoxin. Fine. In that case, obviously each S is coming from a cysteine residues. So the first statement is actually a true statement. We have to identify the incorrect one, that means the false one. But the first one is a true statement. Let us look at the second statement. It is talking about 2Fe2S. That means you have to remember the structure for Fe2S2. What was the structure for Fe2S2? Let me write it down here. We have S, this is another S. Now we have S, this is cysteine, this is cysteine, this is cysteine, this is also attached with cysteine. So in case of Fe2S2, we have 4 sulfur atoms that are part of cysteine residues and to another H2 minus ligands. So overall we have 6 S donors. That is correct. In case of structure of Fe2H2, you can see we have 1, 2, 3, 4, 5, 6. 6 S donors. Out of these 6 donors, these are the H2 minus ligands. They are not part of cysteine. They are directly H2 minus, and the other sulfur atoms, other 4 sulfur atoms are part of cysteine residues of protein. So yes, the second statement, statement B, is also a true statement. Third statement is a 4Fe4S pyridoxine contains a cuban core. So let us look at the structure for that. So this is the structure for 4Fe4S. We can see 4 Fe centers and 4 sulfur centers. Obviously the core of this, it does look like a cube structure. So that's why it is known to have a cuban core because of its cubic structure. So yes, 4Fe4S pyridoxine definitely has a cuban core. So the third statement is true as well. Now looking at the fourth one, it states in a 4Fe4S pyridoxine, 4 redox couples that makes use of the 4 epicenters are accessible in nature. Now these are the 3 couples, redox couples, that we can see in nature. The first one, where we have 3 Fe3 plus 1 Fe2 plus, that can get reduced, resulting in the formation of 2 Fe3 plus and 2 Fe2 plus. That again gets reduced to 1 Fe3 plus and 3 Fe2 plus. These are the redox couples that are accessible. So how many there are? There are only 3. So this statement which says there are 4 redox couples that are accessible in nature is actually a false one. We can only get these 3 redox couples. Okay. So our fourth option was originally a incorrect or false statement. Okay. So I hope this was clear. Let us move to the question number 6. In question number 6, they say the given figure represents the structure of ATP. We all know now that this is the structure of ATP. Hydrolysis of ATP is the catabolic reaction processed by which chemical energy that has been stored in the high energy bonds in adenosine triphosphate is released by splitting these bonds. For example, in muscles, by producing work in the form of mechanical energy. So this is just a whole essay. You just need to identify ATP hydrolysis involves the hydrolysis of which bond. You have the structure here for ATP. Now among all these bonds, you have to identify which bonds gets hydrolyzed in case of ATP during the production of energy. There are 4 options. The first one is the phosphoanhydride bond linking the beta phosphate to the alpha phosphate. Next one is any of the phosphorus hydroxyl group bond, okay, on the terminal gamma phosphate. Third is the phosphoanhydride bond linking the gamma phosphate with the beta phosphate, and the D is bond linking alpha phosphate to the adenine. First let us marking to the phosphate groups. That's how it will be easier for you to understand. So this is your alpha, this is beta, this is your gamma phosphate bonds. So which bonds they are talking about? One option is this bond linking the beta and the alpha one. One bond they are talking about this. This is the option A. Another one is any of the phosphohydroxyl bonds. Third option is gamma to beta. In option number A, they are talking about this bond. Option number C, they are talking about this bond. And in the fourth option, they are saying the bond linking the alpha phosphate to adenine. That means this bond. So this is your option number D. So choose which of them gets hydrolyzed. You already know that. Try to answer it and then I will show you. Okay. Any of you? Okay, fine. So let us look at the option. The correct option should have been option number C. Why so? Because if you remember, we read that already. In case of hydrolysis, if a H2O molecule or suppose a HO minus is going to attack, where the attack will happen? This one, sorry, this nonsense, this one, this beta position, this will come back and this will leave. This portion just discard it. So the attack happens in this bond, that is between your beta and gamma phosphate group. Once this OH minus or H2O molecules attack here, you have your resonance followed by release of this phosphoric acid group, and this other part, this will get cleaved and this other part will be stabilized through resonance. So that is what we learned in case of ATP hydrolysis. So the correct option should have been option number C, the bond between gamma and beta. And again, so after you have the hydrolysis at this position, so you are left with one P11O, O minus, OH, OH. Another part is you have your ADP. One phosphate group has left, so now you have your adenosine diphosphate. Again, if H2O molecules have to attack here, then it will attack the bond between alpha and beta phosphate group, because now we only have two, alpha and beta. So once it attacks the alpha position, that will again release another phosphate group and you will end up with AMP or adenosine monophosphate. Okay. So I hope this is clear. Let us move to the question number 7. This is a very easy one. All of you should know this one. What is the balanced equation half reaction for the reduction of nicotinamide adenine dinucleotide from NAD plus to NADH, that is the reduced form of NAD plus? What is the balanced half reaction? Select your option, then I will show. Out of these four, which is the balanced half reaction for the reduction of nicotinamide adenine dinucleotide? Even if you do not remember, just by looking at the reaction, you would be able to understand what is the balanced equation. So, what do we have here? One second. Yes. We are going from NAD plus to NADH. How that transition will go? What do you require to go from NAD plus to NADH? You require a H plus, because you can see we have added an H plus here. So obviously you require an H plus for this transition. Now we have to balance this equation. We know this, that NAD plus will get reduced in presence of H plus to give you NADH. But now we need to balance this equation. How to do that? Now see, how many positive charges do you have here? H plus and NAD plus. And how much charge you have on the right hand side? In left hand side, we have two positive charges. In the right hand side, what is the charge? Can you see any charge on NADH? No. So in the right hand side, we have 0 charge. Fine. Now we have to adjust the charge as well. How to do that? We have 2 plus here. That means we have to add 2 minus charges on the left hand side, so that overall we get a 0 charge on the left hand side as well. That means how can I do that? If you have 2 electrons here, that means the overall charge gets neutralized. You have NADH on the left hand side as atoms if I considered. Okay, although the NAD is a big molecule, just I am considering that you have your NAD and H, all of those atoms on the left hand side. Similarly, you have NADH on the right hand side. On the left hand side, you have two positive charges. Now you have added two electrons, that means overall charge is 0, and also on the right hand side your overall charge is 0. So that should be the balanced half reaction for this reduction. So which option matches with this? Obviously option number D. So option number D should have been the correct answer for question number 7. Okay, let us move to the next one. It says a flavoprotein contains one FAD moiety. If this is the only electron acceptor in the flavoprotein, how many electrons can the flavoprotein accept when taking on its fully reduced form? So you have your flavoprotein, that means it has flavin. So there is only one FAD that is involved in the overall reaction. Now you are fully reducing FAD. So how many electrons can this flavoprotein or the FAD associated with flavoprotein can accept during this process? First of all, you need to understand that if FAD gets reduced, what it will form. Try to answer it and I will show that in a minute. Write your answer in the chat box and I will show the correct one in a minute. Try to answer that because I think all of you do know it. Do know the answer. So I have written down the structure for riboflavin that is associated with FAD. So you have to identify the reduced form of it. Okay, fine. So what should be the correct answer? FAD does get reduced. I am not writing the whole structure. If it gets reduced, so you have the ring in the middle, this 6-member ring in the middle. I am just not writing the other substituents, they are still there. So FAD gets reduced at this position and this position. Let me just 1 second. Just let me draw here. So after reduction you have one hydrogen atom here and another hydrogen atom here. That means two hydrogens are involved in the reduction of FAD. These two positions gets reduced. So once you reduce FAD, you will end up with what? FAD. Two hydrogens are there now. So you have your FADH2. So the conversion is FAD to FADH2. Now just like this, so this is the fully reduced form. If half reduction would have occurred, we will first end up with, so FAD, let me write it down, FAD can reduced in a half form to go to FADH. That means only one of these two position will get reduced, and in the next step it can get fully reduced to FADH2. This is the overall process. Now in this question they are talking about the fully reduced form. So we have our, let me write it down, fully reduced form means FAD to FADH2. They are talking about this form. Now it is asking how many electrons can flavoprotein accept while this reduction that is going on. That means we basically need to balance this reduction reaction, half reduction reaction. Okay. So what you have in your left hand side, you have your FAD and here is FADH2. So what do you require for reduction? 2 hydrogens are added in the right hand side. So you also need to add 2H plus on the left hand side to balance it out. So in both the sides now we have FAD and 2 hydrogen. Now let us look at the charge, just similar to the previous question. On the left hand side we have 2H plus, that means 2 positive charge, but on the right hand side there is no charge, the overall charge is 0. So to balance out, what do we require? We also require to add 2 electrons on the left hand side. Now overall this will be neutralized, so on the left hand and both right hand side will have 0 charge. So that is the balanced equation. So how many electrons are getting involved in this process? 2 electrons. So the correct answer for question number 8 should have been option A. Okay, I hope this is clear. Let us move to the next one. So this is again a very easy question, but you have to read through all the lines. So it states NAD and FAD are important molecules for electron transport in our cells. We have already read about them. We have talked about NAD as well as FAD. NAD exists in two forms. One is the oxidized form, that is written as A. One second. Yes. So what is, let me change the color to this. Yes. So what is our A? A is the oxidized form of NAD. Okay, you have to identify how do you denote it. Then it is saying, and the reduced form is denoted here as B. So the reduced form is B, that is reduced NAD. Again you need to identify B. Then it states, whereas FAD exists as C in its fully reduced form. So what is C? So our C is fully reduced FAD, not the half reduction but fully reduced FAD. So you need to identify that as well. And also it is saying the difference between NAD and FAD in terms of electron transport is that FAD transport n number of electrons. How many number of electrons they have not said. So how many electrons does FAD transports, that you need to identify as well. Okay, unlike NAD which transports only one. Okay, so these are the 4 things that you need to identify: A, B, C and D. So A is the oxidized form of NAD. So what should be the correct answer? What is the oxidized form of NAD? It is NAD plus, the plus sign. Okay, so let me write it down. So the oxidized form of NAD is known as NAD plus, obviously. B is the reduced form of NAD. What is the reduced form of NAD? That is NADH. C is fully reduced form of FAD. So what is the fully reduced form of FAD? FADH2. And the fourth one is how many electrons gets transport through this process? That is 2 electrons. Just in the previous slide we discussed that 2 electrons gets transported. Fine. So this question was very easy. All you had to do is read the statements that are given and identify which species they are talking about. Otherwise, the question was altogether very easy. So matching with this, the correct option should be option number D. A is your NAD plus, B NADH, C is FADH2, and N, the number of electrons, is 2. So let us move to the next one. It says cytochrome C oxidase contains all except one of the following. Which is the odd one? So there are 4 statements. They are saying out of these 4 statements, only one is the odd one. That means one is incorrect statement. So you have to basically identify among the 4 ones that which one is the false statement. That is, you need to identify. So the options are given. Go through them, write your option in the chat box, and then I will discuss the statements one by one. Try to write your answer, try to guess. So the first statement is a CO2 center bridged by 2 cysteine residues. Second one is a heme unit with 2 axial histidine units. C is a copper center coordinated by 3 cysteine residues, and the fourth one is a heme unit with 1 axial histidine residues situated nearest copper center coordinated by 3 histidine residues. These are the 4 statements. Okay, so let us look at the structure of cytochrome C oxidase at first. Okay, so on this top corner what you can see is the structure of cytochrome C overall. Okay, now dividing them in 2 parts, I have shown it here. If you divide them in this 2 part, this part is shown here and the heme part another one. This is your heme part. Okay, the 2 structures are shown here in individual manner. Okay, now let us look at the options one by one and identify them. So the first one says a Cu2 center breached by 2 cysteine residues. Now let us come to the Cu2 centers. So how many copper centers we have? We have 1 and 2. So yes, 2 Cu centers are there. And what are they breached by? What these 2 copper centers are breached by? There is cysteine 196 and 216 in the left hand side, and on the right hand side we have cysteine 200 and 220. That means copper 1 and copper 2 are actually bridged by cysteine residues. How many? 2 cysteine residues. Fine. So yes, the first one is a true statement. Coming to the next one, it is saying a heme unit with 2 axial histidine units. Actually it exists, because if you think this is the heme unit that you are looking, only this planar part, this black planar part only you are looking. Now look at this red circle, that is your iron center. Now this iron center, you can see there are 6 dashed lines. 4 are planar, that is due to these porphyrin rings, and another 2 on the axial position. And these axial positions, now look at this structure below. On the axial position, actually if you place them according to this structure, then histidine here and histidine here. These two will fall under this coordination on the axial position. So yes, the heme unit on the axial positions has two histidine units. So absolutely option number B or the statement B is also a true statement. Coming to the third one, it is saying a copper center coordinated by 3 cysteine residues. We already know that this statement is false, because if we look at any of the copper centers, we can see there are 2 cysteine residues that it is attached to. The other is methionine and histidine. So every copper units, in case of cytochrome C, we have 2 cysteine residues and 1 methionine residues and 1 histidine residues. So the 3 cysteine residues is a wrong statement. So our fourth statement was actually the false or incorrect one. Still, let us look at the last one. It is saying a heme unit with 1 axial histidine residue situated near a copper center coordinated by 3 histidine residues. So again, we have our heme unit with we know that in case of heme units, we have 2 axial positions which are covered by these histidine units. I have already marked them. Again, they are saying that there is a third histidine residue, that they are situated near a copper center coordinated by 3 histidine residues. Is it true? Yes, this heme moiety does have 2 histidine residue on its axial position, but the copper center that they are coordinated with has, each of them has single histidine residues connected to them. Just look at copper 1 here. Copper 1 has 1 histidine residue attached to it, coordinated to it, and copper 2 also has 1 histidine unit attached to it. So this part of this statement, that the copper centers are coordinated by 3 histidine residues, was a wrong statement. Sorry, I think this should be 2. There is no, this one should be actually 2. The statement otherwise would be false. You can clearly see the copper N is also connected to single histidine unit, and this one is also connected to single histidine residues. Okay. So let us move to the next one. Fine. So question number 11 is saying, what is the terminal component in the electron transport chain that reduces oxygen to form water and that can be poisoned by cyanide? So you have to identify the terminal component for electron transport where you get oxygen to water. So you have your 4 options: NADH dehydrogenase, cytochrome oxidase, succinate dehydrogenase, and coenzyme Q. Select your option and then I will show it to you. So where do you have our oxygen to H2O conversion? That only happens in case of complex 4. So now if you do remember, we have 4 complexes, complex 1, 2, 3, 4, that were involved in case of the, that is required for the synthesis of ATP. We have 4 complexes that are involved, and out of those 4, only complex 4 is involved in the reaction where you get oxygen to form water. If I write it down here, the correct form would be, just let me just write it down here, 2 cytochrome C reduced plus 4 H N plus H2O2 to 2 cytochrome oxidase 2H E plus H2O. So that was the reaction that was happening in case of complex 4. Out of these 4 reactions, complex 1, 2, 3 and 4, in the fourth one we have our, this one is, I think this one is looking like H2O2. Let me write it down. This is half O2. Just let me cut it down. This is basically half O2 that I was trying to write. Okay. So this is the reaction that was involved in the conversion of oxygen to H2O. So you can clearly see which one or which terminal component is involved here. It is cytochrome oxidase, you can see here. So the cytochrome oxidase that is involved in case of complex 4 for transferring this reaction from O2 to H2O. Now the last statement that is says that it can be poisoned by cyanide. What does it means? That whenever you have probably you have heard about cyanide poisoning or carbon monoxide poisoning, etc. So what happens basically in case of cyanide poisoning, the cyanide can bind strongly with the cytochrome oxidase, and if cyanide binds, what happens, the oxygen molecules will no longer bind to your cytochrome oxidase because something else is already sitting at this position. That means your cytochrome oxidase is now unable to take up the cellular oxygens and do the necessary function that is required for your ATP synthesis. That means the whole ATP synthesis will stop in your system. So that is what happens in case of cyanide poisoning. So although this was not related to it, the question, but you should know that cytochrome, this cyanide poisoning is actually occurs due to the tight binding of cyanide ions to cytochrome C. It is also known as the histotoxic anoxia or hypoxia, where the cells cannot bind to the oxygen molecules anymore. Okay. So this was the answer. Let us move to the next question. This is a very easy one. I think all of you know it. So please do answer this question in the chat box. I definitely believe all of you know this answer. So what is the relationship between delta G and delta G0? What is the reaction or relationship between delta G and delta G0? So you have your reactant to product. So how you will write the reaction? RT ln what? Please select it because I know all of you know this question. This is the very basic equation. At least one of you. Come on, these ones are you all of you know. Anyway, so what do we write? Delta G is equal to delta G0 RT ln product by reactant. You have to be very careful about the sign. If you are writing the equation at delta G0 minus RT, then obviously the equation will be reactant by product. But if you are using this positive sign, the product will be on the upper part of this division. Okay. So the correct option for question number 12 should have been A. Delta G is equals to delta G0 RT ln P by R, or products by reactants. Okay. So let us move to the next. Fine. This is a very important question, mathematical question. All of you please try to solve it by yourself and tell me the answer that you are getting. I will help you, I will tell you how to solve it, but please try it by yourself as well. So in this question they are saying, calculate the delta G for the creation of an electrochemical gradient of protons in a mitochondria when the membrane potential is 150 millivolt and the pHs are 7 and 8 on the P side and the N side of the membrane respectively. Now they have given the values of R, T and F as well. You have to calculate the delta G value. So what do we have here? So this was our outer membrane for mitochondria. If this is your cristae and this is the inner membrane, so it is the intermembrane space, that is otherwise known as the P side. And here, because this is the overall structure, that will be the structure of mitochondria, I am just doing the one part. So inside you have your matrix, that is otherwise known as the N side. So what is the pH at this P side? It is 7. And what is the pH at N side? It is 8. Everything is given, and due to the difference in the proton ions that exist between this intermembrane space and matrix, that's why we have positive charges along this membrane and negative charges along this side. So this difference in charge leads to a membrane potential. This membrane potential is 150 millivolt. Now you have to calculate the delta G. Please try it out by yourself, and then obviously I will show you the solution. So what do you have here? You have to calculate delta G. For that, the equation is 2.303 RT delta pH plus ZF delta psi. So you already have this equation. You know the value of R that is given. You know the temperature value. What is the delta pH value? Delta pH is nothing but the difference between this P and N side. So it is the pH difference between N minus pH difference at P side. Obviously, because at the P side you have more number of H plus, I should have written P, N minus P. So because at the P side you have more number of H plus, that's why the pH will be less at that side, and on the N side you have low number of H plus, so the pH will be higher. So delta pH is pH N minus pH P. You already have the values for those. And what is Z? You already know the charge that is associated. So you are just passing out H plus atoms, protons. So that means the Z here associated with it is 1, because these are only single positive charge you can see, and F value is already given, and delta psi is your membrane potential, that is the potential difference along which you need to take the protons or pump the protons. So delta psi is your membrane potential, or the potential difference along the two sides of this membrane. So membrane potential, that is also given in your question, that is 150 millivolt. So put all the values in this equation and tell me what answer you are getting from it. Okay. Please do the mathematics yourself at least. I have shown everything, given you the values. Just solve it out and tell me what value you are getting. Put everything here. Delta G equals to 2.303. R is your 8.315, that is given in your question. T is 298. Delta pH is pH N minus pH P. pH in is 8, pH of P side is 7, so our delta pH is 1. So we have our delta pH 1 plus, so this is one of the values, plus we have our ZF delta xi. So Z is 1, F is 96,485 coulomb, third is constant, and you have your delta xi 150 millivolt. Now make sure you have converted everything to the same units. So everything here we are calculating in coulomb into volt, that means joules. So here also I will convert this 150 millivolts to volt. So that will be 10 to the power minus 3. This is coulomb, this is volt. So ultimately that would result in coulomb into volt would result in joule. So if you do this calculation, I still do not see any answer here. So if you just solve this simple mathematics, you will end up with an answer of 20.2 kilojoules per mole. That will be the answer. Something around like 20.17 kilojoule per moles or something will come. That will end up to 20.2 kilojoules per mole. So please, even if you do not say it here, try to solve it out by yourself. Later, the correct answer should come 20.2 kilojoules per mole. Okay, fine. So let us move to the next question. So it's shown it. So question number 14 says glucose 1 phosphate can be converted. One second. Glucose 1 phosphate, yes, it is showing. Glucose 1 phosphate can be converted to fructose 6 phosphate in the following two reactions. So there are two reactions that are given. One is glucose 1 phosphate to glucose 6 phosphate. So that is the intermediate, and again glucose 6 phosphate converts to fructose 6 phosphate. And the corresponding delta G0 values are also given there. So the value of delta 0 for the overall reaction you need to, or for the overall conversion you need to calculate. This is also an easy one. Try it out and then I will show. I hope you all are able to solve the question even if you don't say the answer here, because these are very easy and similar questions will come to the exams as well. So what are these reactions? Let us just see. So we have our glucose 1 phosphate. Let us name as A. So we have our conversion from A to B. Let us name glucose 6 phosphate as B. Now B is again being converted to fructose C phosphate. Let us name it as C. So what are these reactions? You can see this is a coupled reaction. Why? Because we have a similar intermediate in between them. So in case of this is our first coupled reaction, half coupled reaction, and this is another half of this coupled reaction. So this is the first half of this coupled reaction. This is the second half of this coupled reaction. So how do you get the delta G value in case of this coupled reaction? By adding the individual ones, the delta G value of these individual half reactions, you can get the delta G value for the overall coupled reaction. That's what we learned in the class. So what should be the value of this overall reaction? A to C. So basically you need to calculate the overall delta G for the reaction A to C. So how we will calculate it? We will calculate it by delta G of A to B and summing it up with delta G for B to C. That is what we learned. So let us see what is the delta G value for the reaction of A to B. It is minus 7.1 kilojoules per mole. And what is the delta G for B to C? We have our minus 1.7. So overall the unit is kilojoules per mole. So what should be it? It should be minus 8.8 kilojoules per mole. So that should have been the answer. So what matches with it? Option number D. Option number D. Be very careful, we had a negative sign in the front because option number B is same but it does not have the negative sign in front of it. Be very careful while doing this mathematics about the sign because minus or plus just changes the overall spontaneity of the reaction. Just by forgetting a minus sign in the front, you can change a spontaneous reaction to a non-spontaneous one. So be very careful with the sign in the front. Okay. So option number D should have been the correct answer for question number 14. Let us move to the next one. We have this, the overall reaction of complex 4. Just a few slides back we discussed about the reaction of complex 4. The complex 4 reaction can be summarized in the caption below. Provide the missing coefficients as N, M, X, Y, Z. So what did we learn? Just a few reactions back we did this. Cytochrome C, I even wrote the overall equation. So you have to identify N, M, X, Y and Z. So let us write it down here again. So we have our 2 cytochrome C reduced that reacts with 4HN plus that reacts with half of oxygen and gives you 2 cytochrome C reduced plus 2H plus at the P site. Let me write it as capital, that would be easier for you to understand, and 1 H2O molecule. So that is the reaction. Just a few slides back we have discussed this. Now let us look at the option. For this particular equation that we have written, the values of N, M, X, Y, Z should have been 2 followed by 4 followed by half followed by 2 followed by 2 and this is 1. How we are getting it? We are just looking at the balanced equation. Here you can see we have 2 cytochrome C, 4HN plus, half O2, 2 cytochrome C reduced, 2H plus at P site and 1 H2O molecule. But none of the options matches this one. Okay, you can see out of these 4 options none of them matches this one. Why not? How can we match it? Just see in the some options here we have 1/2. We do not want any kind of decimal, so let us just multiply this by 2. So the overall reaction will remain same, the overall balance is remaining same. We are just multiply the overall balance that we have done with 2, but ultimately it will remain as the same reaction. If we do that, what we will end up with? We will end up with 4. Here we will have 8 followed by we will have, here something is written as in Fe2 cytochrome C. Yes, fine. So it should be 1, it should be 4, it should be 4, and it should be 2. I think the only this option should be 481442. Yes. So I think in this particular one, this one should be the answer, but the third option should have been 1. This is given wrong. See, if we multiply it by 2, we can clearly see the answer is coming as 2 into 2, it will be 4 cytochrome C plus 4 into 2, that will be 8 HN plus. Instead of half oxygen, we have now half into 2, that means 1 oxygen. Obviously 4 cytochrome C reduced to 4 cytochrome C. Here it will be, sorry, I wrote it wrong, it will be oxidized. Then from 2H plus it will be multiplied by 2, ending up with 4, and lastly it will be H2O. 2H2O it will be ultimately. I think this one was this should be the correct option, option number A, where the third digit was actually given wrong. So the correct option should be 48142. Okay. Hope this is clear. Let us move to the next question. It says the biochemical reaction A to B has a K equilibrium value less than 1 under physiological condition. So you have K equilibrium less than 1. Which of the following best describes the delta G parameter of the same? So how do you relate delta G with K equilibrium? We know delta G equals to minus RT ln K equilibrium. Now this K equilibrium value is less than 1. That is what is stated in the question. Now if you put a value in the ln that is less than 1, you will end up, this ln K equilibrium will ultimately end up giving you a negative value. So if this ln Ku now becomes negative, you already had a negative side initially, so ultimately that will give you RT some value that we don't know. So overall the sign becomes positive. That means delta G has, because of K equilibrium is less than 1, delta G is positive. Obviously so, because K equilibrium less than 1 means that equilibrium is not spontaneous. That equilibrium will not go forward. That is also evident from the delta G sign as well. You have a positive delta G value in that case. So delta G, obviously if you multiply R will be say like 8.3 for something. If you take the normal temperature 25 degrees centigrade, that is 298. So and some value of X will be there. After multiplying everything, obviously overall the value will be very greater than 1. So the correct option for question number 16 should be option A, that in case of K equilibrium less than 1, delta G becomes very greater than 1. Let us move to the next one. Fine. So this one states ATP synthesis is attained by coupling the free energy of a proton gradient to the chemical synthesis of ATP. The enzyme that accomplishes this coupling is what? So there are 4 options. So from the name itself you can already guess. So basically we are dealing here with synthesis of ATP. So what should be the enzyme that is related to it? ATP synthase. From the name itself you can identify, because we already know the different role these different types of enzyme has. Lipases, lysases, oxygenases and syntheses all have different roles. We have already learned this in some other lectures. So obviously in case of synthesis, the synthase enzymes will take part. So the correct option for question number 17 is ATP synthase. Okay. Let us move to question number 18. It is saying when the delta G0 of the ATP synthesis reaction is measured on the surface of the enzyme ATP synthase. So we have our enzyme that is involved, ATP synthase. Now you are measuring the delta G0 on the surface of the particular enzyme. It is found to be close to be 0. Delta G0 is found to be close to 0. Why this is happening like that? Why the delta G0 for ATP synthesis on the surface of the enzyme comes around 0? Delta G0 is 0 means that is a very good type of equilibrium process that is going there. Why that is happening? So these are the 4 options that are given. First one is a very low energy of activation. Second one is enzyme induced oxygen change. Third one is stabilization of ADP relative to ATP by enzyme binding. And the fourth one is stabilization of ATP relative to ADP by enzyme binding. So these are the 4 options that you need to see. So first of all, a very low energy of activation, that is not the thing, because you do require the proton pump to occur to simultaneously transfer the H plus ions and couple that reaction with ATP synthesis to get your overall delta G as a negative. That means the overall activation is low. That is not true. The enzyme induced oxygen change is not responsible for this part as well. Out of this third and fourth statement, you know, in case of ATP synthesis, we start from our AMP that goes to ADP and that goes to ATP. So finally, we are actually bothered about this final ATP, the energy source that we require. Obviously, if that is the final product that we desire, that should be tightly bounded to the enzyme. So obviously the synthesis of ATP, after it forms, it is stabilized relative to the ADP with the enzyme. That means the 3 negative charges are much more stabilized by these enzymes, by some other groups of enzymes that are involved, but it stabilizes ATP to a much more extent compared to your adenosine diphosphate. That is why obviously the reaction will go forward. If the ADP binding is very much favorable, then the next step obviously will not happen. That is very obvious. If ADP binding to enzyme is very much favorable, it will not try to convert it to ATP. But because ATP is heavily stabilized by the enzyme binding, we get to have this conversion from ADP to ADP, thereby resulting our desired molecule. So the correct option for question number 18 should have been option number D. Let us move to the next one. It says, which of the following electron carriers is not able to transfer one electron at a time? So we have 4 options: NADH, FMN, FAD and ubiquinone. These are the 4 options. Now if you do remember, in case of nicotinamide, let me just draw it here. I don't think I have the structures. Okay, so just let me draw it here. So we had our nicotinamide, CONH2, and this is our nitrogen site. That gets, I am not writing the other part, there are other parts involved. We have our 2 electron to 2H plus that end up in resulting this nitrogen. We have our 2 hydrogens attached here, and that is our CONH2. This is known as our NADH. This was our NAD plus. This was our NAD plus. That results in, let me just write it down as R. This is your R as well. So this was your NAD plus that gets reduced to NADH. Only if you follow the mechanism of this reduction of all these things like NAD plus to NADH, FMN to FMNH, FAD to FADH2 and ubiquinone to the reduced form of ubiquinone, that is the QH2, basically Q to QH2. In all those transfers you can see, although NADH, FAD and coenzyme Q, all of them can transfer to a hydrogen, but in case of NADH the transfer happens in a single step. It is not like the case of FAD. Like if you remember, in case of FAD we can go from FAD to FADH, that ultimately again can get reduced to final reduced form FADH2. That means two different process can happen. Similarly we can get from Q to QH and as well as QH2. But in case of NAD plus, the direct conversion happens to NADH. It is not able to carry the electrons or transfer the electrons one at a time. Both the electrons, both the protons, the reduction all happens in a single step. So the correct option for question number 19 should be option number A. Moving to the next one, it says reduced QH2 is not formed by which of the following. So again we have our four complexes, complex 1, complex 2, complex 3 and complex 4. For that you need to remember which complexes involve which of the enzymes and which of the cofactors. Now regarding that, just let me write it down. So in case of complex 1, yes. So in case of complex 1, I think there was a table. Still let me write it down. Complex 1, we ultimately end up from, the reaction is NADH, 5H plus are getting transported from N side. With the help of Q, NADH gets transferred to NAD plus. We have our QH2 and 4 protons go to the P side. That is the reaction for complex 1. So in case of complex 1, we get reduction of Q from QH2. So the option number 1 is not correct, because here they are saying reduced QH2 is not formed, but we can see in case of complex 1 we have the formation of QH2. Let us look at the complex 2. Complex 2 actually is not involved in any kind of proton transfer. Still, in case of complex 2 as well, if we write it here, in case of complex 2 as well, we have the conversion of succinate to, let me write it down, to fumarate, while the Q gets, coenzyme Q gets transformed to coenzyme QH2, that is the reduced form. So again complex 2 and cytochrome C, that is also supports these reactions, does not end up with a oxidized form of Q, but it also results in the formation of CoQHT. Complex, I think, sorry, this was, I wrote for complex, one second, very sorry, this was for complex 2. So complex 2 was wrong option. I have written for complex 2. Now let us come back to complex 3. The reaction for complex 3, if we write it down, QH2 to cytochrome C that are in Fe3 plus, plus 2 hydrogen from the N side, they go to formation of Q, plus 2 cytochrome C, Fe2 plus, and you have your 4H plus on the P side. So if you look at it, just let me turn back to red. Yes, if you look at it, only in case of complex 3 we can see the conversion of the reduced form of coenzyme Q to oxidized form of coenzyme Q, that is written here. So that is very obvious, because each of the cofactors that are getting reduced or oxidized throughout the reduction throughout the process has to go back to its original form. So in case of CoQ as well, or Q, because its original form, the oxidized form that is written as Q, it was present at the beginning, but through the reactions of complex 1 and 2 it is getting transferred to QH2, that needs to again be converted to Q for the first two steps to happen. If it stays in this reduced form, then after some time the chain of the reactions will no longer occur. For that you need the reoxidation of this form, that happens through that happens through complex 3, where we can see the conversion of QH2 to Q. So the correct option, that is reduced QH2, is not formed in which of the reaction? In case of complex 3 involving cytochrome C. We can see in the reaction here that we get the oxidized form of Q, not the reduced form of QH2. So if you do remember all the equations of 1, 2, 3 and 4, you will able be to answer that in case of complex 3 we do not get the reduced form of QH2. Moving to the next one, it says if electron transfer in tightly coupled mitochondria is blocked with antimycin A between cytochrome B and cytochrome C, then what will happen? Now again, in case of the overall process, if you go through all the reactions of complex 1, 2, 3 and 4, especially in case of complex 3, you can see, if you see the cycle, you will be able to see that both cytochrome B and cytochrome C are involved in case of complex 3 reactions. Now it is saying, if you somehow block this reaction between cytochrome B and cytochrome C, then what will happen? If you block their conversion that is happening, that one is getting oxidized, another is getting reduced, if you just stop this cycle, ultimately the complex 3 part will not the reaction will not progress any further. That means at complex 3 reaction it will fail. This will no longer progress. What does that mean? If complex 3 reaction does not proceed, you will not have your complex 4 reaction as well. That means eventually the overall chain of the reactions will stop, because ultimately by adding all those equations you will get your final equation for ATP synthesis. By adding all the equations that you get, you will be able to synthesize ATP. But hampering any of the steps or any of the electron transfer reactions will result in complete stoppage of the ATP synthesis, because these all reactions are coupled to each other. If any of them stops, obviously the other will no longer function. So if you block cytochrome B and C in the mitochondria with antimycin A, then ultimately it will lead to the stopping of the ATP synthesis overall. Okay. So let us move to the next question. It states the relative concentration of ADP and ADP control the cellular rates of what? So we have our ATP and ADP, the adenosine triphosphate and diphosphate. What are the processes they are basically involved in? Now, obviously we know the first is glycolysis. We know that we require ATP to hydrolyze glucose molecules. Glycolysis means that, why we require that? Because Ultimately, the hydrolysis of glucose will result in the energy in the form of ATP that we require. That means the concentration of ATP and ADP is very important in this case. So glycolysis is an important thing that is involved in this reaction. In case of ATP and ADP, concentration is very important for glycolysis. The second one is oxidative phosphorylation. Oxidative phosphorylation, we are learning this actually. Oxidative phosphorylation means the process of the synthesis of ATP through all the electron transfer processes. Obviously, in case of oxidative phosphorylation, as we are dealing with the synthesis of ATP, the concentration of ATP and ADP will be very important in that case. So again, oxidative phosphorylation is heavily dependent on the concentration of ATP to ADP to control the cellular rates. Then comes pyruvate oxidation. So here I am showing the pyruvate oxidation. What you can see, this pyruvate is getting oxidized to acetyl CoA. In the process, we have the conversion of NAD plus that is getting reduced to NADH. What does that mean? We are getting more number of NADH. Where this reduced NADH will get used? Again, because once you are converting to reduced to oxidation, again you have to perform their oxidation to reduction. That is the cycle. One thing you are reducing, ultimately you have to take it back to its oxidized form again in case of all those cofactors. So where this reduced NADH will be used again? In the ATP synthesis. So of course, for pyruvate oxidation as well, the concentration of ATP and ADP will be very important. The only fourth option, you can say respiration, that is not directly related with the concentration of ATP and ADP. So the cellular rates that are dependent on ATP and ADP concentration are 1, 2 and 3. Except for all the other 3 processes are very much dependent on them. Moving to the next one, it says a student proposes a probable enzyme kinetics that is shown here, wherein he proposes the half reaction. So we have 2 reactions. This is our first reaction, half reaction. This is our second half reaction. And the corresponding delta G values are written as well. You have to actually calculate the delta G0 for the overall coupled reaction. So what is the overall coupled reaction? You just simply add them. If you add these 2 equations, what you will end up with? You will end up with, on the right hand side you have A plus B. Now you are adding the another equation, so x plus y, that is giving you S plus y, B plus Z. Now these 2 and these 2 are same on both the sides, so we do not require them. So ultimate equation is A plus x2 S plus Z. So by simply adding them, we can get our overall coupled reaction. So how to calculate the overall delta G as well? So overall delta G would be delta G of reaction 1, the first half reaction, and delta G of the second half reaction. So what are the values? Plus 31 minus 14. So if you do that, you will end up with 17 kilojoule per mole. Obviously 17, there is a positive sign in front of this. That means delta G is positive. So the reaction is energetically unfavorable. So what should be the correct option? That is delta G's value will come as 17 kilojoule per mole and the overall reaction is energetically unfavorable. Moving to the next question. So this is basically the structure of flavin adenine dinucleotide structure of flavin adenine dinucleotide derived from vitamin riboflavin is shown. So this is basically talking about, one second, this is basically talking about FAD. State whether it acts as an electron or proton acceptor or donor. So you all know FAD gets transferred to FADH2. Ultimately it gets reduced to FADH2. So In that process, we already know FAD, we have 2H plus 2 electron, that will result in the formation of FADH2. So it does acts as a 2 electron and 2H plus acceptor, because by accepting these 2 hydrogen and these 2 electrons, it is resulting in the formation of FADH2. So it is a 2 electron 2H plus acceptor. So the correct option for question number 24 should be option B. Moving to next one. So it states a student synthesizes the enzyme whose structure is shown above. Considering that A is the protein component, so this part is our half spherical part is our protein A, and B is this Fe2 plus ion. State what are the two components are commonly known as. This question we have learned a lot ago, because long ago we have learned about this question. We already know that overall we call this type of compounds as holoenzyme, where we have our protein part plus non-protein part. We have already learnt it in previous lectures. The protein part, that we call apoenzyme, and the non-protein part, depending on whether it is tightly bound or covalently bound or loosely bound or non-covalently bound, we can classify them further as coenzymes or prosthetic group, cofactors and prosthetic groups. So in case of non-protein part, we can see the Fe2 plus generally remain as coordinated, that is they are tightly bound to the protein centers by some coordinate bonds. That means in this particular case, as it is tightly bound, we will call this non-protein part as prosthetic group. Also, as a metal ion is involved in this case, we will call it a prosthetic group. So what we have here, this protein part is our apoenzyme. So that means A is our apoenzyme, this option, and B is our prosthetic group. Together we will call them holoenzyme. So the correct option should have been option number A. Moving to the next one. So it says the given scheme shows the mechanism for an enzyme action. Name the enzyme. So this is the reaction that is happening, that is shown on the right hand side. What we can see in this reaction, what is happening, this is basically the cleavage of this amide bond that is happening here. So let us look at the option. First option is oxidase. Whether it is an oxidation reduction reaction is going on. No, we do not see any kind of oxidation or reduction reaction. This is just a bond cleavage of this peptide bond, right? So either oxidase or reductase is incorrect, because from the name itself we can tell oxidase and reductase means they are involved in some kind of oxidation and reduction reaction, but none of this is happening in this. So the first two options are incorrect. And the fourth option, if you see, it says amylase. Amylase is involved for the cleavage of glycosidic bonds, right? Do you see any kind of glycosidic bonds here that are present in case of sugars? First of all, these are not sugar moieties, you do not have your glycosidic bonds. So amylase here will not act upon them. The only option remain is hydrolase. Is obvious because you can see the involvement of a H2O molecule. So the H2O molecule is basically that is attacking here, resulting in the cleavage of this amide bond. So the enzyme that is involved here for this reaction is nothing but your hydrolase. Okay. Moving to next one. This is saying identify the correct statement regarding the structure depicted here. So the structure that you are showing, what is this? It is one subunit of your ATPase, right? ATPase had two subunits. One is F0 F1, because that is why it is known as the overall ATP synthase is otherwise known as F0 F1 ATPase. That is because it has two subunits. One is, or two main components. One is F0 complex and another one is F1 complex. The image that you are seeing here is the image for F1 complex, right? So it is F1 ATPase. In this question we have to identify the correct statement. So the first statement where it says it is a F1 ATPase, it is a true statement. Second one, it says the given enzyme generally consists of 5 subunits. Absolutely, the F1 complex have 5 subunits: alpha, beta, gamma, delta and epsilon. Okay, these are the 5 subunits in case of F1 ATPase. We can already see you have your alpha, beta, alternative alpha and betas. You have your gamma. This rotation happens around this gamma unit. You have your delta and epsilon units here. So second statement is a true statement as well. And the third one says the 5 subunits are generally in the stoichiometric ratio of this. They have given the stoichiometric ratio. This last one should be delta. So this is the stoichiometric ratio that they are showing. But if you do remember, the original stoichiometric ratio was 3 alpha, 3 beta units, 1 gamma units, 1 delta units and 1 epsilon unit. These are the ratio for them. So we have 3 alpha, 3 beta, that is making this bird like structure. You have your gamma 1 along which the rotation of them will take place, and you have 1 delta unit and 1 epsilon unit. That is all you have. So the third statement was actually a false statement where the stoichiometric ratio was wrongly given. So the correct statements are 1 and 2, and accordingly the correct option should have been option number A. Moving to the next one. It is also a very easy one, just you have to fill the blanks. So in this they are saying, in a biological system, when a spontaneous reaction may drive a non-spontaneous reaction energetically. So basically you are coupling a spontaneous reaction with a non-spontaneous one so that the overall reaction can occur spontaneously. So what this process is known as? We know this process is known as energy coupling. This is known as coupled reaction. So they are known as energy coupling. Fine. Next thing is, changes of such reactions generally are what? So how do we get the energy change in case of coupled reaction? Simply by adding them, their individual delta G values are added to get the overall delta G. So free energy changes of such reactions are generally what? They are additive. The third one is, certain enzyme catalyzed reactions are interpreted as -1. Two coupled half reactions, because you know in case of certain enzyme catalyzed reaction you have an intermediate. We have done this as a mathematical form as well, that you have the formation of an intermediate and that intermediate can further take up another reaction giving you the final end product. In those cases you add those half reaction and get your full conversion. So two coupled half reaction in case of certain enzyme catalyzed reaction can be done to get the overall spontaneous reaction. So they are known as two coupled half reaction. Okay. So this is C is your two coupled half reactions. So what should be the, accordingly what should be the correct option? The correct option should be option number D. Energy coupling, they are additive, and in case of enzymes they are known as two coupled half reactions. This one state, question number 29, it says, in the biological systems, electrons are transferred from one molecule to another, basically from donor to acceptor, in 4 different ways. The addition of carbon compounds by the utilization of dehydrogenases occur by what mechanism? So if you do remember, in case of this electron transportation, 4 processes were there. Either the electrons can directly get transferred, or they can be transferred in form of hydrogen atoms. Third one was they can be transferred as hydride ions, and fourth one was direct reaction with oxygen. These were the 4 ways by which electron transportation can occur in biological systems. So in the first cases where you have involvement of dehydrogenases, they generally occur by hydrogen atom transfer. So you have your FAD or NAD. So in those cases, or say suppose succinate dehydrogenase, in that cases if you see, you have the transfer of hydrogen atoms involved. So dehydrogenases generally occurs by hydrogen atom transfer reaction. So our one is hydrogen atom transfer. Second is NAD linked dehydrogenases. The first one was normal dehydrogenases like succinate dehydrogenases. Second one is NAD linked dehydrogenases act by what? What mechanism? So if you remember the structure, how it converts from NAD to NADH, you will remember there is a H minus that gets transformed to one of those carbon centers. That means it is a transfer of a hydride. So NAD linked dehydrogenases act by hydride transfer mechanism. So our 2 should be hydride transfer. And in case of oxygenase, it is very obvious from the name itself, oxygenases will react by directly interacting with oxygen. So 3 should be direct reaction with oxygen. So these were the options for question number 29. I think this is the last one. Question number 30, it says the depicted dimethyl isoalloxazine ring system is known to confer certain degree of planarity to a certain vitamin, regarding it a yellow coloration. The vitamin is in discussion is what? Already from this structure you should have identified what we are talking about. So what is this structure? Where you have seen it? In case of riboflavin, in case of FAD you have seen it. From riboflavin we can get to the FAD. So riboflavin is this dimethyl isoxazoline ring. This ring system is known as this dimethyl isoalloxazine ring, that is known to confer certain degree of planarity and gives some yellow coloration. So that vitamin is riboflavin, which is otherwise in short form known as vitamin B2. Okay, so I think that was it. Yes, so that was the last question of this session. Okay, so fine, we will meet next in 16th April, I guess. So you can post your queries in the SWAM portal. For now, I will end the session and you all can leave now.